go to question number 1. Potato tissue or grapes become flaccid when immersed in A. Isotonic solution, B. Hypertonic solution, C. Hypertonic solution, D. Cold water. When we take any biological tissue like a potato tissue, or you take a cell of grape, when you take the grapefruit, the cell, and you take the potato tuber or the grape cell which has got a semi-permeable membrane, and you immerse it in a solution which is primarily hypertonic. Now, when you say hypertonic solution, it means excess salts. So, when you immerse a biological cell, the biological cell by nature is hypertonic, and when you immerse it into a solution which has got higher concentration, there is always flow out, which is termed as exo-osmosis. That is, here we have, when you take the grape solution or the potato solution, the total amount of the solute in the solvent will be less, whereas in a hypertonic solution there is more concentration of the solute per solvent. Therefore, here because the solute is less, it will have more of free energy. So, from a region of lower free energy, it goes to another region that is with higher concentration. So, this process is called exo-osmosis, and during exo-osmosis, the biological cell will turn flaccid. The meaning of the term flaccid is it can collapse or it shrinks. The process is termed as exo-osmosis. So, when you take a tissue and immerse it in a higher concentration, the cells will shrink and they will show exo-osmosis. So, among the choices, the answer, if it will shrink normally when it is placed in a hypertonic solution, hence the answer is B. We go to question number 2. Symplast means A. out of free space, B. the continuity of plasma membrane and continuous living inner space, C. the adjacent cell walls and intercellular spaces, D. the space of water movement by free diffusion. Whenever we take any biological cell and we are dealing with relation to transport of water as well as organic food materials, there are two important concepts. One is termed as symplast and another is termed as apoplast. The total of the living cells, all the living cells, including the continuity of the plasma lemma or the plasma membrane, which forms a continuous living space and connects all the living cells of a plant. This inner space is termed as a symplast, whereas when you take the free space outside, the free space which is outside the cell wall, which includes the intercellular spaces and the vacuoles, this is termed as apoplast. Now, apoplast primarily relates to transport of water. The same can be illustrated in a diagram. When you take a biological cell, the outer space, we have an outer space so this space is termed as apoplast. The apoplast is made up of the outer space. It includes the plasma membrane, intercellular spaces plus vacuoles. Whereas when you take the symplast, you got the living cell, you have the plasmodesmata, and again there is a continuity of cell number 1 with cell number 2. So, a continuity of living cells with the plasmodesmata is termed as a symplast. Water moves via the apoplast. So, when you say symplast, it primarily means the choice B, the continuity of the plasma lemma and the continuous living inner space. Hence, the right answer is B. We go to question number 3. The pathway of absorbed water is soil water, root hair, and the choices are A, cortical cells, passage cell, pericycle cell, xylem, leaves. B is passage cell, endodermis, phloem, leaves. C, cortical cells, pericycle, xylem. D, cortical cells, passage cell, xylem, phloem and leaves. Whenever we think in terms of the path of water in plants, it predominantly starts with the soil water. We have the soil water. From the soil water, it enters the root hairs. After it enters into the root hairs, from the root hairs, it passes through the cortical cells. Firstly, it passes through the cortical cells. That is, prior to this, it goes through the epidermal cells. We have the epidermal cells, then we have the cortical cells, then we have the endodermis. The endodermis has specialized cells which are termed as passage cells. Via the passage cells, it reaches the next tissue which is termed as the pericycle. After the pericycle, the water enters through the vascular bundle, especially it enters into the cells of the xylem, which is the principal water conducting tissue in plant cells. And from the xylem upwards, it moves into the sort of a leaves. So, the pattern of path of water includes first is soil water, second is root hairs, then it passes through the cortical tissues, via the endodermis or the passage cells, through the pericycles, into the xylem and later on into the leaves. When we look at the choices, the path of absorbed water is we have choice A which says cortical cells, passage cells, pericycle cells, xylem and the leaves. Hence, the choice A is right. Question number 4. Within the root hair cells, which events are sequential? A, there is less water, TP, turgor pressure, more OP, osmotic pressure, increased DPT, diffusion pressure deficit, high suction pressure, endoosmosis. Then B, due to A, exoosmosis only occurs. C, due to A, the OP, SP, DPT becomes lower and TP is increased, that is turgor pressure is increased. D, there is more water, high TP, loss of OP and decreased DPD. Now when you take root hair cells, which are involved in water absorption, we have a typical root hair cell which is supposed to be taking in water from the outside. Now, a typical root hair cell initially to start with has less of water, and because it has got less of water, it also has got less of turgor. Now, once water enters in, its osmotic pressure increases, and with increased osmotic pressure is indicated by the presence of increased diffusion pressure deficit plus dPt, and also there is high suction pressure. Now, because there is higher suction pressure, it results in endo-osmosis, that is, water from the outside gets into water on the inside. Hence, the path of water uptake shows all these particular features. Now, parallelly, as a consequence of this, the osmotic pressure OP, then again the suction pressure, as well as the diffusion pressure deficit, and all these three become low after some time, and thereby what happens is the turgor pressure is increased. So, we have at first level all these features. At the second level, the osmotic pressure, the suction pressure and the diffusion pressure decreases, and thereby consequence of this, lastly we have the turgor pressure is increased. Hence, among the choices, if you look at choice number C, answer C, which says due to A as well as other sort of consequences, because of this primarily in the root hair cells water gets absorbed. Hence, the right answer is C. Question number 5. Aquaporins are A. associated with the osmosis process, B. water transport channels in cell membranes, C. water pores, D. phosphorus rich water. When we take aquaporins, aquaporins are protein molecules which are present in the plasma membrane. So, these molecules are primarily termed as aquaporins. They are embedded in the plant plasma membrane. So, the aquaporins are nothing but proteins. They have gates for entry of water, and they have been discovered as the basis for symplast, that is living space, symplast transport of water in plants. So, they are membrane bound proteins. They are membrane proteins which help in transport of water, especially through the sort of a symplast. So, among the choices, we primarily have the choices. Answer is B. They are water transport membranes. Question number 6. A cell becomes turgid when placed in A. A is isotonic solution, B hypertonic solution, C hypertonic solution, D ice with salt. When you take a typical biological cell, a typical biological cell is rich with relation to solutes, ions and other enzymes. So, because it is primarily rich, it is hypertonic. A biological cell is very much active. So, when it is placed in a hypertonic solution, that is a solution with lower concentration of solutes in the solvent. So, because water has got more of free energy, water tends to move inside. The movement of water, especially with relation to water, movement inside the biological cell is termed as endo-osmosis. And when there is movement of water, the cell becomes turgid. Turgidity is due to the presence of a turgor pressure. So, we have a turgor pressure as well as we also have an osmotic pressure, and at a later period the osmotic pressure becomes equal to the turgor pressure. When OP is equal to TP, then diffusion pressure deficit becomes 0. So, till that period of time the water keeps on going via the process of endo-osmosis. So, turgidity of a biological cell is attributable when it is placed in a sort of a hypertonic solution. So, among the choices, a cell becomes turgid when placed in B, that is a hypertonic solution. The answer is B. Question number 7. Water needs to move from cell to cell. This is due to A, OP, osmotic pressure, B, TP, turgor pressure, C, imbibition, D, DPD or diffusion pressure deficit. The prime reason why water movement occurs in biological cells, especially from cell to cell, cell A to cell B, is attributable to one very important property called diffusion pressure deficit. Now, diffusion pressure deficit is the variation in the pressure for diffusion between the solute and the solvent. The differences in the ability of the solute and the solvent to move across a biological cell is expressed in terms of diffusion pressure deficit. Now, DPT can also be calculated in terms of OP and TP. So, whenever the diffusion pressure deficit it becomes 0, till that particular time osmosis primarily occurs. So, the entire movement of water movement in plant cells is attributed to one central principle which is termed as DPD. Hence, the answer is D. Question number 8. Potometer works on the principle of A, potential difference between tube and the plant, B, amount of water absorbed equals that transpired, C, root pressure, D, osmotic pressure. When you take a potometer primarily exactly measure how much of amount is taken in and how much of water is lost via transpiration. It is shown in a simple sort of an experiment which is done in the lab, wherein we have a tube and one end of the tube immerses into a solution and the other end we have a sort of a cut plant. So, the cut plant continuously transpires and the one end of the particular plant takes in a lot of water. So, the differential amount of water absorbed versus the amount of water which is transpired. The differences between these is primarily the principle of the potentiometer, which is a typical classroom measurement of both the amount of water absorbed as well as the amount of water transpired by a sort of a living plant. Hence, the answer is B. Question number 9. The values of dPd and water potential are A plus and minus, B minus and plus, C both are plus positive, D both are negative. When you take the values of diffusion pressure deficit as well as water potential, now diffusion pressure deficit is normally measured as a positive value and water potential is measured as a negative value. Diffusion pressure deficit primarily indicates the difference in the pressure which can enable diffusion between the solute and the solvent. Water potential primarily ascribes to the amount of work done. Potential means the amount of work done in depending upon the free energy of water in a biological system. Now, normally when you take the water potential of free water is 0. Now, whenever water has got to move, it is beyond free water, thereby its value is always negative. So, water potential is always treated as a negative quantity, whereas diffusion pressure deficit is always treated as a positive quantity. Hence, among the choices, the choice of answer A is right, which indicates diffusion pressure deficit is plus, and again water potential is given as primarily in the form of a negative value. Question number 10. Water potential in plant cells and tissues is given as A, W is equal to S, B, water potential W is equal to S plus P, or C, water potential W is equal to dPd plus S plus P, or D, W is equal to M plus S plus P. Now, water potential is usually given a sort of a symbol like psi. So, the psi of water potential is always given in all living cells and also in all tissues as a combination of the psi of the matrix potential, which is a term which is used for dry cells, along with the potential of the solute potential, the capacity of the solutes to do work, and it is a combination of the pressure potential which is exerted on the cell walls. So, water potential as a sort of a complete, this is the definition which has been given by Slater. So, according to Slater, water potential is a combination of the matrix potential as well as the solute potential and the pressure potential within living cells or tissues. So, when you say water potential, it refers to matrix potential. Now, matrix potential primarily is a term which is relevant to how much of matrix is present, or the biological matrix which is present within dry cells. Like if you take the seeds, as well as it also is an indication of what is the solute potential, what is the capability of the solute to move in a solvent. Depending upon its potential, the capacity to do work, the amount of the solute molecules which is capable of doing work, as well as how do you calculate it in terms of pressure. That is, once water moves into a biological system, the water can exert pressure on the cell walls. So, when it exerts pressure on the cell walls, it leads to turgor or turgidity. So, we have a turgor pressure. Now, parallel to the turgor pressure, because water moves into biological cells, we also have an osmotic pressure. the turgor and the osmotic pressure in turn build up a sort of a wall pressure. So, a combination of all these three is also taken into consideration along with the solute and the matrix potential, and we have the sort of a general equation for a water potential in plant cells and tissues. So, among the choices, answer D, which is a complete statement, is right. Question number 11. The stoma opens when the guard cells swell due to A. Loss or decrease of water potential, B. Increase of water potential, C. Endoosmosis and efflux of H plus ions, D. Endoosmosis because of influx of protons or H plus ions. When we have the stoma is the pore, and the stoma is surrounded by guard cells. These are the guard cells, and guard cells in turn are surrounded by the subsidiary cells. So, guard cells plus stoma is equal to stomata, and stomata are the units of transpiration in leaves. Now, whenever the stoma or the pore opens, it opens due to swelling of the guard cells, that is, the guard cells become turgid. The turgidity of the guard cells is termed as swelling, and swelling is attributable to the accumulation of water. Turgidity is attributed to accumulation of water, and the accumulation of water, especially in the guard cells, if you take the guard cells, this is attributable to one very important basic function, which is termed as the increase in the water potential of the guard cell. When the water potential, that is the amount of free energy which the water is needed in order that it get transported between the guard cells and the subsidiary cells, whereby there is increase in turgidity and the stoma can open, is attributed to the water potential. So, initially a high water potential, when you say a high water potential, it means the more of free energy is required for the transport of water. So, high water potential in guard cells is due to the accumulation of water, which leads to turgidity, and turgidity of the guard cells in turn determines how the stoma pore can open or close. So, the opening of the stoma is attributed to the guard cells. Hence, among the choices, the right answer is B. Question number 12. In a fully turgid cell like a guard cell, choices are A. OP is equal to TP, so DPD is 0. B. TP is 0, so DPD is equal to OP. C. DPD is greater than 1. D. DPD is less than 1. When a cell is fully turgid, it means the osmotic pressure is the highest. When the osmotic pressure is highest, the osmotic pressure will create a sort of a wall pressure. There will be a WP which contracts the OP, and when this sort of a contraction occurs, it results in the formation of a TP, turgor pressure. When osmotic pressure is equal to wall pressure, or wall pressure is equal to turgor pressure, under these sort of conditions, the diffusion pressure deficit becomes 0. That means the cell is fully turgid. When the cell is fully turgid, water will not enter or leave the system. Thereby, it is said to be a sort of a turgid cell. So, the word here is fully turgid, that is, there is no exodus of water or there is no influx of water. There is no influx, there is no efflux of water. So, with relation to water, if there is no influx or efflux and the cell is fully turgid, it means OP is equal to WP or TP and DPD is equal to 0. So, under these sort of conditions, the choice A works out right, hence the answer is A. Question number 13. Cut beetroot slices in water do not enable the water to turn pink due to A. Plasma membrane being impermeable to anthocyanin, B. Anthocyanin being more in water than in beetroot, C. Anthocyanin being bound to gums, D. Plasma membrane being permeable to anthocyanin. Whenever we take beetroot, and you slice them, any beetroot slice in water when it is put in water, we have beetroot slices, and when they are placed in water, then the water does not turn pink even when you heat. The reason it does not turn pink is primarily when you take the beetroot cells, the cells have got plasma membrane, and the plasma membrane is impermeable to anthocyanin. That is, the anthocyanin or the red pigments which are present in beetroot, they cannot penetrate the plasma membrane or they do not break the plasma membrane of the beetroot cells. Thereby, the red color does not efflux or red color does not come and cause colored water. Hence, among the choices, the answer is A. So, this is one experiment to prove that the plasma membrane, biological membrane which binds cells, and it is selectively permeable. It only allows certain ions to move through it. In this case, it does not allow the passage of the anthocyanin pigments into the sort of water, thereby the water remains colorless. Question number 14. Solution A has a semi-permeable membrane with OP, osmotic pressure, of minus 30 bars and turgor pressure of 5 bars. If it is submerged in a solution B with OP of minus 10 bars and 0 turgor pressure, the direction of net osmotic moment is A to B, B to A, C 0 moment, D equal moment in both the directions. Imagine we have two solutions, solution A and solution B. Which are separated by a semi-permeable membrane. Now, in solution A, the osmotic pressure is OP is equal to minus 30 bars and the turgor pressure is 5 bars. Whereas in solution B, the OP is minus 10 bars, it is minus 10 bars and the turgor pressure is 0. Now, primarily the differences between the OP and the TP, that particular value gives the value of the diffusion pressure deficit. So, here in this case it is minus 35, you get is 25. And here you got minus 10, 0, you get 10. Now, this is minus and this again is minus. Now, normally the direction of net osmosis is always from a higher value to a lower value, hence the direction is always from A to B. Among the choices, the right answer is A. So, we need to take the turgor pressure value and deduce it from the osmotic pressure value and assume the particular value, and looking at the directions, we know the direction of how osmosis proceeds. Question number 15. Solution A has a water potential of minus 9 bars and solution B has minus 4 bars. They are separated by a semi-permeable membrane. The direction of movement of the water will be A to B, B in both directions, C no movement, D, B to A. When you take solution A and again solution B, which are separated by a semi-permeable membrane, we understand that solution A, the water potential is minus 9 bars, and in solution B it is minus 4 bars. Now, these are indicators of the water potential, that is the free energy of water. The more negative the value and the more higher the value, this is a higher value which is a negative value, it means the water potential here is extremely less. And here it indicates the water potential is extremely more, that is minus 4 is greater than minus 9 in terms of water potential. Thereby always it moves from a value which is higher, that is from minus 4 to minus 9, hence the direction of movement of water is from B to A in terms of the water potential. Hence, the right answer is D. So, we need to primarily understand if the value could be higher, but the negative value indicates that its value is actually less. Question number 16. The lower the water potential, the A, it means water stress, B, the harder is its movement, C, greater is the ability of the water to be absorbed by plants, D, lower is the ability of water absorption. The lower the water potential, it means the greater is the ability of the water to be absorbed by the living plants. Hence, here among the choices, the right answer is C. Question number 17. Guttation is caused by A, transpiration, B, osmosis or DPT, C, OP, D, root pressure. Guttation means the loss of excess water. If we find dew droplets during early mornings, when there is more of mist, this process of liberation of the waste water is termed as guttation. So, guttation primarily relates to excess liquid exudation, and it is a passive process. It does not require ATP and it does not involve the stomata, and at the same time it does not cause any sort of a cooling of the plant tissues, and it is a passive mechanism. How does guttation differ from transpiration? Transpiration results is an active process. It involves the stomata. It causes cooling of the leaf, and there is only loss of water in the form of a vapor. Whereas guttation is a process wherein excess liquid is lost in the liquid form in a passive mechanism, non-involvement of stoma, and does not cause cooling, and it is primarily attributable to root pressure. So, exudation of excess water is attributed to pressure at the level of the roots. Hence, the answer is D. Question number 18. A leaf peeling of Tradescantia is placed in 10% sodium chloride NaCl. Under observation in a microscope, after 10 minutes, we notice A, endoosmosis, B, cell shrinkage, C, separation of the protoplast from cell wall, D, both B and C. So, when we take a leaf cell of Tradescantia, Tradescantia is a very favorable plant wherein you take the epidermal peelings, wherein you can easily see that what happens to cells. So, if we take a leaf tissue, place it in a petri dish, into which you primarily add 10% of sodium chloride. Now, sodium chloride is primarily a salt solution. Now, when you take Tradescantia leaves, if you take the Tradescantia epidermal cells, the epidermal cells would primarily shrink. The cell would shrink. The shrinkage is termed as imbibition, and here there would be the filling of the isotonic solution. So, surrounding a cell which has shrunken or which shows endo-osmosis, the cell shrinking is called endo-osmosis. There is the separation of the protoplast from the outside cell wall. So, we have a cell wall. Next, we have an isotonic solution in between. Next, we have the protoplast, and the protoplast shrinks. The gap between the protoplast and the cell wall is filled by the isotonic solution, which is neutralized when it is put in sodium chloride. Thereby, when you look at the choices are B, there is cell shrinkage, and C, separation of the protoplast from the cell wall. Hence, the answer is both A and C are correct. Choice of answer D, which says both B and C are correct. Question number 19. Dry seeds placed in a beaker of water swell. This is due to A, absorption, B, adsorption, C, diffusion, D, imbibition. When you take dry seeds, especially if you take cereal seeds or you take pulse seeds which are dry and you place them in a beaker of water, the seed coat can take in little bit of water because it is hydrophilic, because they are rich in colloids, and it causes adhesion of particles, and whenever there is adhesion of particles, that is the water gets into the sort of a seed coat, the seed coat can crack, and we find that the dry seeds, they show break in the cell walls. The same is true with relation to doors during the rainy season. Doors will primarily crack. So, this process of taking in water by the process of adhesion due to the presence of colloids is predominantly termed as imbibition. Now, imbibition is a passive process when compared to osmosis. It is a passive process. Hence, the right answer is D. Question number 20. Which is an antitransparent? A. Cobalt chloride, B. Phenyl mercuric acetate, C. Potassium iodide, D. Mercury. An antitransparent is a compound which prevents transpiration. Like if you take leaves and you rub them with phenyl mercuric acetate, PMA. PMA is a liquid. It is also used as an aerosol, which when put on leaves, it forms a coat which acts like an anti-transparent. So, it prevents excess loss of water. And thereby the plant tissues tend to remain fresh. So, PMA could primarily have an sort of an application if you are transporting apples, or if you are transporting any particular fruit, wherein on the fruit wall, if an anti-transparent is sprayed, there is no fruit respiration. Thereby the tissues remain fresh, and it primarily is a method of post harvest prevention of transpiration and respiration. So, a common anti-transparent which is used in the case of plants is PMA or phenyl mercuric acetate. Cobalt chloride is a paper which primarily turns red to purple, indicating transpiration due to water loss. Potassium iodide is again a sort of a test which is primarily used in photosynthesis, and mercury is primarily a sort of a heavy chemical for plants. Hence, the right answer is B, PMA. Question number 21. If a cell with DPD 4 bars is connected to cell B, C, D, whose OP and TP are 4 and 4, 10 and 5 and 7 and 3 bars, the movement and flow of water will be A, C to A, B and D, B, B to A, C and D, C, A to D and B and C, and D, A to B, C and D. We are taking a cell A. Now, if a cell A with DPD of 4 bars is connected to another cell B, cell C, and again cell D, whose OP and TP values are 4 and 4. Then we have 10 and 5, and later on we have got 7 and 3 bars. The question asks, how is the movement and flow of water? We understand in this case the DPD value in cell A is 4 bars, and when you take OP minus TP, when you apply the principle of OP minus TP, OP minus TP for cell B is 0. Hence, always we understand the diffusion pressure deficit occurs from a region of higher DPT to lower DPT. Hence, part of the question for the answer is it moves from A to B. Then again, depending upon cell C and cell D, we understand in this case the DPD is 3, and in this case the DPD is 4. But however, when we take into consideration in the form of bars, this is a negative value and this is a negative value. Now, minus 4 bars is a lesser value, minus 3 bars is a more value. Thereby, always whenever we take the flow of water, the flow of water is again in this case also it is from C to D. Hence, the correct answer is choice B. Question number 22. A cell is extremely turgid. One of the given values indicates 0. A. Suction pressure oblique DPD oblique water potential. B. Osmotic or solute potential. C. Turgor pressure. D. Wall pressure. We primarily understand when you take any particular cell, the meaning of the term extremely turgid would always imply that the value of diffusion pressure deficit is equal to 0. Now, whenever the diffusion pressure deficit is equal to 0, that is OP is equal to TP is equal to wall pressure is equal to 0, or there is no differences between OP and TP, then also the suction pressure parallelly also becomes 0. So, in this case, when you take the water potential value, it also tends to be 0. So, in a fully turgid cell, the attributes are, first important attribute A is DPD 0, B thereby suction pressure is 0, and C we also have the water potential is also being 0. Hence, among the choices the answer is A. Question number 23. The most common method of water absorption involves A. Osmosis has a main role and is called passive absorption. B. Negative pressure created by the xylem sap. C. Occurs continuously and meets total water requirement of a plant. D. All the above. Whenever we are dealing with water absorption in plants, the total water absorption in plants is predominantly determined by the process of osmosis. That is the selective movement of the solute oblique by a solvent via a semi-permeable membrane. The process of osmosis is passive. It is by passive absorption, that is, it does not involve metabolic energy like ATP. ATP is not involved, and it is created because of a negative pressure. The negative pressure is attributable to the xylem sap. The sap gives in a sort of a negative pressure or a pull, and the process of water absorption occurs continuously. It occurs continuously, and all of these together determine the water requirements of plants. So, the total water requirements of plants is due to a continuous water column which is developed under negative pressure of the xylem sap, and it is a passive movement. It may not involve metabolic energy, and the principal process is osmosis. Hence, among the choices we have the right choice is D, which includes all the above, and the sequential of the events include the process, the phenomenon is by the process of osmosis, and mostly it is passive, it is continuous, and it is under negative pressure. Question number 24. Passive absorption relates to A. Lack of use of energy and mass flow. B. Root cells are inactive. C. Both A and B are correct. D. Roots which are active and ATP from respiration is used. Whenever we are dealing with passive absorption, passive absorption means there is non-use of ATP, of adenosine diphosphate, or non-use of any particular metabolic energy. And again, passive absorption occurs by mass flow, that is N mass A, total flow of water. and predominantly here the roots are inactive, they are not actively involved. The roots are inactive, and here when you take, if roots are active and primarily they involve the ATP, it includes the symplast movement, whereas when you take the apoplast movement, primarily in apoplast we have passive absorption. So, any non-use of ATP by mass flow wherein the roots are inactive is attributed to the passive absorption of water, which includes the choice answer is C, which says that both statement A and statement B are correct. Question number 25. Water movement, if active absorption occurs, means A. movement against chemical potential gradient ATP, B. free space and apoplast, C. symplast, D. both A and C. If we believe in the presence of active transport, of water, if active transport of water need to occur, it need to move against a chemical potential gradient. That is, if you take a cell, in the cell there are several chemical reactions against the chemicals. If there is a sort of a potential created, there is variation in the charges and a gradient is created, the gradient is against the chemicals, so it is termed as against the chemical gradient. Within the cell, and if it involves ATP, as well as it can occur primarily in the free space. Then you call this particular thing as primarily a method wherein there is active transport, and the choice here is both A and C, hence the answer is D.